Shall I start?
Yes.
Yeah. Ma'am will start.
Sir, I will start.
Okay.
Heartily good afternoon and warm welcome to all delegates. I, Mrs. Ashwini Shelke, Assistant Professor, Department of Pharmaceutical Chemistry at Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, Nashik. Welcome you all to first session of Day 4, 1 week international level e-FDP, e-STP, entitled Recent Advancements in Pharmaceuticals, Academic Research Ethics and Social Perspectives, organized under aegis of IQAC MSG Copper. I specially welcome to Dr. Prashant Shinde sir, our first resource person for today's session. Before we begin with our session, I would like to introduce him. Sir is a research scientist in analytical development laboratory process, Glenmark Life Sciences Limited, Mumbai. Sir has completed his PhD in pharmaceutical sciences from ICT, Mumbai. Sir has completed M.Pharm from Government College of Pharmacy, Amravati. Sir has completed B.Pharm from SSDJ College of Pharmacy, Nashik. He qualified entrance for UGC SAP in 2010. He also qualified GATE with All India rank 342 in 2006. Looking at his work experience, Sir is currently working as a research scientist and team leader in analytical method development at Glenmark Life Sciences from 2022. He worked as a senior research officer at Glenmark Life Sciences from 2018 to 2022. Then he worked as a research officer at Glenmark Pharmaceutical from 2015 to 2018. He got a senior research fellowship at ICT from 2012 to 2014. He also got junior research fellowship at ICT Mumbai from 2011 to 2014. He also worked as an assistant professor at ST College of Pharmacy, Shirpur from 2009 to 2010. Then looking at his awards and achievements, Sir has got UGC SAP fellowship for doctorate in from 2011 to 2015 and master fellowship of UGC from 2006 to 2008. He successfully delivers analytical methods for different APIs. He performed installation of new Empire server 3.6.1 with IQ, OQ, PQ and installation of new thermo IC instruments. He won a Supernova Awards for the method development of Favipiravir. He has 11 publications in national and international journal of high reputation. So let's take advantage of his expertise. I request to Dr. Prashant Shinde sir to start his session on nitrosamine impurities with regard to current regulatory environment. Please, sir. Over to you, sir.
Okay. Good afternoon, all. So today I will delivering a lecture on nitrosamine impurities and its implication in current regulatory environment in pharmaceutical formulations. So I first like to thanks EMS Gosavi College of Pharmaceutical Education and Research to given me opportunity to deliver session and then share my thought on this topic. Okay. So first question is here I am considering that all the viewers here are from the academic background and many of them are students also. So I am covering a basic of nitrosamine here in this presentation, today's presentation. So we'll get to know about this new happening in pharmaceutical industry. This topic generally started coming in discussion from the 2018. That history we will going to discuss in this presentation also. And from 2018 onward, it is an highly discussed topic in pharmaceutical industry, nitrosamine impurities and its control, how to control it, how to analyze it, and there are several severe implications if nitrosamine are present in your formulations or in your APIs. So I think as a pharmaceutical personnel or people working in pharmaceutical industry or wish to work in pharmaceutical industry should know what is the nitrosamine impurity. So first is, what is the nitrosamine impurity? It is a simple answer. In organic chemistry, nitrosamine impurity, or more formally it is called as N-nitrosamines, are organic compound with the chemical structure R2N N double bond O, and where R is usually an alkyl group. To simplify this, this is an next is the structure. Now you can understand this one. If this functional group is there, N double bond O, and alkyl functional group attached to it, we called it an nitrosamine imprint or nitrosamine compounds. These nitrosamine compound are known from ages, you can say, but it is its presence in pharmaceutical are discussed from the start of 2018. Usually, this nitrosamine can be found in food also. It is a food contaminant. It is generally found in potable water also, or it can be formed by grilling of the meat also. But in pharmaceutical, it is recently has been discussed. This compound can be formed by nitrosating reaction between amines. Amine required for this reaction to form is secondary amine, tertiary amine, quaternary amine. Primary amine does not undergo this reaction, and it react with the nitrous acid to form the nitrosamine impurities. For this reaction to occur, generally this reaction in pharmaceutical occur with the sodium nitrite reagent. That sodium nitrite get converted into nitrous acid in the presence of acidic environment, and that nitrous acid react with the secondary, tertiary, and quaternary amine to form the nitrosamine impurities. So this is a representative reaction how these nitrosamines are formed. These are nitrous acid react with the secondary amine to form this in situ intermediate, and then give rise to the nitrosamine. Now question is, why we are discussing the nitrosamine? Because this chemistry is well known to everyone, I think, but why we are discussing this nitrosamine, that is the question. So what happen is, if this nitrosamine are present, this nitrosamine get ingested in a by anywhere from pharmaceutical, or by ingested through foods and drinks. So what this nitrosamine can cause, this is a representative figure. This nitrosamine undergoes alpha hydroxylation reaction, and it forms diazonium ion, and then alkylation occurs, and this alkyl group or carbonium ion can cause the cytotoxicity or mutagenicity or carcinogenicity. In simple language, if nitrosamines are present and get ingested frequently, it can give rise to the formation of cancer. It can cause cancer, or it can cause mutagenicity to the cells. So controlling these impurities, that's why important. So again, question is, though nitrosamine are known, now we know about what it can cause if it is present. So obvious question is, why as a pharmaceutical person it is an important thing. So in ICH M7 there is a guideline. In ICH called M7, it talks about genotoxic impurities, this guideline, and it is that ICH M7 consider this N-nitrosamine impurity as a known mutagenic and carcinogenic, and classified them as a class 1 impurity. And it is cohort of concern, means if these impurities are present in your samples, then it is cause of concern, and it may cause cancer to the patient. So again, question arise, though it is an impurity that is well known, why we are still discussing it? That anyone who knows the pattern of pharmaceutical industry knows that if it is an impurity, it should be controlled. So still, why we are discussing this nitrosamine, why it is started from 2018, what I am saying. So in June 2018, FDA was informed, FDA, Food Drug Administration, and we are talking about US FDA. So this FDA was informed of the presence of impurity identified as N-nitrosodimethylamine, which is also called as NDMA, in the angiotensin receptor blocker Varsantan. These drugs is of ARB class, that all you all know. It is certain category drug, generally used in heart failure and all. And it is reported in 2018 that this valsartan drugs contain the NDMA impurity. N-nitrosodimethylamine above the acceptable level. Through investigation, FDA determined that numerous lot of valsartan and few other ARB drug product. ARB means angiotensin receptor blocker drug, like various sartan, losartan, varsartan, RB-sartan, telmisartan, and all from different manufacturer contain unacceptable level of nitrosamine. This announcement lead to voluntary recall. This is an class 1 recall. Generally, there are three type of recall in FDA: class 1, class 2, class 3. Class 1 is more severe recall, and this lead to voluntary recall of hundreds of batches of the generic version by pharmaceutical distributor worldwide. Means any formulation which contain losartan, valsartan, telmisartan has been recalled. Some are voluntary recalled it. Some FDA has directed to recall because of presence of this nitrosamine impurity. Further, FDA and EMA is European Medical Association, investigate in year 2000 led to detection of this nitrosamine impurities in pioglitazone also used for treatment of diabetes, and it is also found to be present in some batches of ranitidine. That is an histamine blocker used for the treatment of acidity of the stomach. And presently also there are reporting of the nitrosamine presence in metformin drug also. So starting from June 2018, you can see there are various drugs which are found to contain this nitrosamine impurity. Before that, it is not reported that any drug containing this nitrosamine impurity. But through some investigation of residual solvent and all, people come to know that many of these drugs are having presence of this nitrosamine. And as you already know from previous slide, that nitrosamines are genotoxic in nature. They can cause cancer if consumed above certain level. So it is a cause of concern from FDA also. That's why FDA recall many of the drugs which found to contain this nitrosamine impurities. So this is again in 2019, Jantac and Histac, which is formulation of ranitidine, found to contain unacceptable level of NDMA, and FDA recommend this manufacturer to voluntarily recall the ranitidine and nizatidine product with NDMA level of above what agency consider as unacceptable. So after that, again investigation has been continued, and in 2020, FDA has decided to completely withdraw the ranitidine product from the US market. Ranitidine, as you know, it is an antacid drug commonly prescribed, but still from the US market, it is completely withdrawn due to presence of this NDMA, because they found consuming the ranitidine causing severe health concern due to presence of this NDMA impurity. So I think now you are get the idea why we are discussing this nitrosamine impurity and why it is an important aspects in current pharmaceutical scenario. Because presence of this can cause impact on business in US region, EU region, and many other countries also. So this is an history, brief history where from it is started in 2018. There is company called Prashant Pharma informed the US FDA about the nitrosamine impurity. They reported that NDMA in valsartan from the API, that is active pharmaceutical ingredient, sourced from the Zhaijing Huai Pharmaceutical, China. Actually, this Princeton Pharma using the API procure from this Chinese company to prepare their formulation, valsartan formulation, and they found out that it contained the NDMA impurities present. They reported it to FDA. Then FDA come into action. They identified 4 different manufacturers using API from the same source and notified them to investigate. So other 4 manufacturers also procuring this API from the Chinese company, and they also notified by the FDA to investigate about the presence of NDMA, as one company has already reported to them that NDMA has been found. Also 20 other manufacturers selling valsartan has been notified. There are other manufacturers also who are taking APIs from other sources, but they are manufacturing the valsartan. That's why they are also notified that the NDMA is present and can be controlled or should be controlled or should be reported whatever level of the impurities this NDMA present in that drug. So in 2018 only, there is voluntary recall of valsartan, that all the product available in market has been recalled by the FDA, and it is not in use at that time. They have directed that anything manufacturer, anything which has been taken from the Chinese company has to be recalled and should be investigated for the presence of the nitrosamine. In the same month, FDA come up with the limit also, that uh, what should be the threshold limit for this impurity to be present in this API, so that it should be an up to the safe level, you can say, or can be consumable. So that limit they have derived from the AIMS test or from the toxicity data available is 96 nanogram per day, and it is based on ICH M7 guideline. So up to nanogram level, it is only acceptable. This calculation is generally based on uh, safety data, and they considered that if 1 lakh people has consumed this nitrosamine impurity, consumed nitrosamine impurities for 17 years up to 96 gram per day, then there is one more cases of the cancer can happen. That is the calculation, and based on that calculation they have decided this limit 96 nanogram per day. Above that, there should not contain any presence of nitrosamine impurity above this level. That is the direction they are given in June 2018. So any manufacturer who have NDMA presence above 96 nanogram per day has to recall the product from the market on immediate basis. That is a class 1 recall. And in class 1 recall, generally there are lawsuits also, and can be filed, and there should be a severe penalty also sometime. So in August 2018, FDA itself has taken, because there are what happened is many manufacturers found that NDMA is present in their valsartan compounds. So they started recalling it, and there is dearth in the market of valsartan. It is an can be considered as a life-saving drug, valsartan, and it is an necessary medication. So FDA has come forward and come up with the analytical method. They published one analytical method based on GCMS to evaluate the presence of NDMA. As you know, the 96 nanogram per day, you can imagine it is at very lowest level. So it require an EMS detection. Normal HPLC and GC cannot able to detect this impurity at this particular level. So FDA has come up with its own method to determine the presence of nitrosamine impurity, NDMA, by using the GCMS method. So in September 2018, there is another impurity has been reported. That impurity is called as NDEA, and it is N-nitrosodiethylamine impurity, and it is also found in a lot of valsartan manufactured by the same Chinese vendor. And import alert in September 2018 has been issued on this company itself. So this company cannot import anything to the US till the matter has been resolved. That alert has been given to this company. So in October 2018, FDA again come up with another method for NDMA detection and NDA detection. Both method are based on GCMS only, but using the liquid injections. So next is in October and November 2018. Other certain up to this level, these 3 months there are only discussion about the valsartan and its presence of NDM and NDMBT. From October November, other sartans also started reporting the presence of NDMA and NDA, like RB sartan, low sartan, valsartan from the Sandoz, Mylan and Teva found to contain either NDMA or NDA. So for the detection of this impurity, NDMA and NDA. Then USFDA inspected this facility, Chinese facility of manufacturing API, and they issued the warning letter to them regarding violation of impurity control, change control, and cross contamination. So they are completely stopped manufacturing of this API company, manufacturing or procuring from this API company. So this is the timeline in brief. First, in 26 in June 2018, NDMA in ZH valsartan found. ZH is same Chinese company which we are discussing. NDMA is found. Then voluntary recall of this valsartan has occurred. Then NDMA also found in other company manufacturer, that is Zhenjing Tiangyu, that also contain NDMA, that is also found. and that is reported by the Taiwan FDA. Then NDA also found in valsartan manufactured by the same Chinese company. Then NDA found in losartan also. Up to this, it is up to valsartan. Then losartan also reported to contain this NTMA impurity. Then NDA also found in irbesartan, other sartan drug manufactured by the Aurobindo Pharma, and it is also can cause it is also cause of concern. So then again another impurity other than NDMA and NDA, NDIPA is reported in valsartan to be present up to 2019. Then NDMA is reported in pioglitazone, antidiabetic drug. Then NMBA is reported in losartan. So this has been started the wheel, I can say, to get to know about the presence of this nitrosamine, various nitrosamine impurities in pharmaceutical formulations. So and it FDA started analyzing all these samples. If it is present, they started recalling all the drugs from the market, and there are severe implications of this presence of this nitrosamine in formulation. So there are various formulation has been started reporting the presence of these impurities. Like here up to this we have seen NDMA, NDA, then NDIP has been found, then NMBA has been found, then NMPA also found in valsartan, then NDMA also found in ranitidine. Yeah, FDA started investigating this ranitidine drug also, and they found that during stability study also this NDMA has found to be increasing. So in 2020, they have completely stopped the selling of ranitidine in US. And this story has been goes on, and there are various along this story there they have published various articles. Like I already told you, they have published some analytical methods. After that, they published some guidelines also to the industry how to control these impurities, what is the mitigation factor, what is the causative agent for this formation of this impurity. And they ask all the manufacturer to comply to this requirement, their requirement. So, how this nitrosamine are formed in pharmaceutical formulation? We already seen how nitrosamine are formed. It requires some nitrating agents and secondary and tertiary amine. But how it is formed in a pharmaceutical formulation? What are the different sources of it forming in a formulation? So, main sources is obviously from the API, active pharmaceutical ingredient, and from the reaction scheme it is formed. So nitrosating agent or precursor used in various route of synthesis are like nitrite ion intentionally used in manufacturing process. As you know, sodium nitrite can commonly be used for the diazotization reaction, and it can be used as a reducing agent for the azides. Nitrite present as an impurity in a reagent. Some reagent contain nitrite as an impurity. This nitrogen oxide can also be used. Nitric acid is used for the reaction. Nitrosyl halide is used for the synthesis of APIs, and even purified water, potable water can also contain some trace level of the nitrosating agent like sodium nitrite. So if sodium nitrite is present and route of synthesis of your drug contain primary, secondary amine, tertiary amine, then it can give rise to the formation of nitrosamine in presence of acidic condition. So these are the source of N-nitrosamine impurity. First is starting material, that is API and its intermediates. Then various catalyst, reagent, solvent, byproduct, and intermediate used. Then various drug substance can also contain secondary amine, tertiary amine, and if it react with the uh, nitrosating agent present in the various excipients, it can also give rise to the formation of nitrosamine impurities. So FDA first identified this 6 nitrosamine to cause potentially a great concern. This 6 nitrosamine is first they identified NDMA. NDMA is N-nitrosodimethylamine. Then NDA, N-nitrosodiethylamine. Then NDIPA, N-nitrosodiisopropylamine. Then NEIPA, N-nitroso isopropylamine. NDBA, N-nitrosodibutylamine, and NMBA, N-nitrosaminobutyric acid. You can see all of this having this nitroso structure, and this all has been found in various APIs and various formulations. And FDA started controlling this impurity through guidelines. They have published some guidelines, they have published some limits, and asked the manufacturer to comply with these limits. So these are various regulatory agencies who worked on nitrosamine and published some guideline to the manufacturer of APIs and formulation. First one is US FDA, which have first time reported this nitrous impurity. Then USP also published some analytical methods to identify to uh, quantify this nitrosamine impurity. European medical agencies, WHO, Health Canada, Brazilian agency Anvisa, and Australian agency TJ, all of them has published some guideline on nitrosamine impurity. So implication of this is, if it doesn't comply with these guidelines, you cannot sell that drug in these various regions. So what are the limit of nitrosamine? So limit of nitrosamine published by FDA. Comprehensive limit has been published in February 2021, but before that also in 2018, 2019 they have published limit on interim basis for this product. So limit for this is FDA recommend following acceptable intake, or you can call the AI in shortcut. Acceptable intake, I already told you, it is depend on uh, 1 in 1 lakh cases of cancer may increase if you consume this above the acceptable intake limit, up to 70 years if you consume these impurities. So these limits are for NDMA, it is 96 nanogram per day. NDA, it is more stringent, 26.5. NMBA 96, NMPA 26.5, NIPA 26.5, NDIPA 26.5 nanogram per day. And as I already told you, AI is daily exposure to the compound such as NDMA that approximate 1 in 1 lakh cancer risk after 70 years of exposure. So they want to nullify the risk of cancer forming due to consumption of this impurity. That's why they have kept the stringent limit for this all the nitrosamine impurity. But this has posed the risk that to identify this impurity at that lower level, at nanogram level, is not possible by conventional techniques, conventional analytical techniques like you are using HPLC, we are using to detect the impurities, or GC you are using to detect this impurity. So it is increasingly impossible to detect at this kind of level. So you have to imply the technique like GCMS, LCMS to identify this impurity. Even FDA has published some methods. They all are based on either GCMS or based on a LCMS. So question is how you calculate this limit while you are testing your drug. So NDMA, I have taken example of NDMA in valsartan. So maximum daily dose of valsartan is 320 mg per day. Then limit can be calculated as AI, that is acceptable intake, which has mentioned in previous table. This table we have seen, and upon maximum daily dose. This calculation you have to do. You get the limit in ppm, at which you need to control the impurity. So based on this calculation, 96 nanogram per day is the AI limit for the NDMA, and 320 ppm, 320 mg per day is the dose of this uh, 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 valsartan. So 0.3 ppm is the calculation which is derived, and at this 0.3 ppm level, we have to control the impurity in formulation or in API also. So FDA has also published table based on various maximum daily dose. There are various acceptable intake they have given, and they have given a calculation also for at what ppm level these impurities can be controlled. And also there is limit for the limit of quantification also, that 0. At 0.03 ppm level we have to give the method uh, having limit of quantification. So this is the guideline what FDA has been published. Though it is February 2021, it is first revision and latest revision. Before that also they have published the same guideline with some minor changes. So highlight of this guideline is first they have given the first is mitigating the presence of nitrosamine impurities in API and how to control these impurities in API. These 2 points they have discussed in brief. There are many others information also, but I have just captured the crux of it. So mitigating the presence of nitrosamine impurities in API, what guideline has been saying that you have to avoid the reaction condition that may produce nitrosamine whenever possible. When not possible, demonstrating that the process is adequately controlled and is capable of consistently reducing the nitrosamine impurities through appropriate, robust fate and per study. So first they are saying that you have to avoid the reaction condition by which the nitrosamine can be formed, and if it is not possible to avoid this condition, then you have to show by appropriate analytical methods that these impurities had been adequately controlled well below the limit of quantification of this particular impurities, and that also you have to show by fate and per study. Using bases, second mitigation factor they said is using bases other than secondary, tertiary and quaternary amines when possible if ROS condition may form a nitrosamine. So they are saying to avoid secondary, tertiary or quaternary amine. If it is not possible, you have to demonstrate through analytical technique that though you are using this secondary, tertiary or quaternary amine, nitrosamine cannot be formed or can be formed within the limit. Using caution when ROS involve the use of amide solvent. If the solvents are present, N,N-dimethyl formamide, N,N-dimethyl acetamide, and N-methyl pyrrolidine, the solvents can give rise to the formation of these impurities. Like NDMA can be formed by N-dimethyl formamide. This N-dimethyl formamide can convert into N-dimethyl amine, and that give rise to the formation of NDMA in presence of acidic condition. If nitrosating agent is present in your route of synthesis. They are also suggesting that you should replace the nitrite and use other quenching agent for azide decomposition process. Optimizing the reaction conditions such as pH, temperature and reaction time so that this impurity formation can be controlled. As you can see, acidic condition is required for the formation of this nitrosamine impurity. So they are suggesting you can work on a pH also so that impurity can be controlled. They are also suggesting to design manufacturing process so that it facilitates the purge of nitrosamine impurities in the subsequent processing step. So if you think that in first step of your reaction nitrosamine can be formed, you have to design the manufacturing process such a way that this nitrosamine impurity in second stage of your ROS can get purged out, and you have to demonstrate that purging through sufficient analytical method. API manufacturer should consider removing the quenching step when there is risk of nitrosamine formation. Example, using nitrous acid to decompose residual azide from the main reaction mixture to reduce the risk of nitrosamine formation. API manufacturers should audit their supply chain and monitor them for any at-risk raw material, starting material, and intermediate. So, any raw material, starting material you are using for the API manufacturing that may itself contain the nitrosamine impurity based on its reaction. So, that raw material should be checked for the presence of this nitrosamine impurity, and you should have sufficient control to purge out this impurity from your process. And also this nitrating agent or nitrosating agent like sodium nitrite can be present in a potable water. So nowadays FDA has given a guidance to check the water used in a process also for the presence of sodium nitrite. So API manufacturer should be aware that potable water used in API manufacturer may contain low level of nitrite and even nitrosamine and environmental contamination. So even nitrosamine can actually be present in this, or from environment it can be contaminated. So potable water should need to be checked for the presence of nitrosating agent and nitrosamine itself while using it API manufacturing. So these are the mitigating factors. Now it is an how to control it. So if nitrosamine impurity is detected above the yellow Q, that is limit of quantification. Limit of quantification is generally 10% of your limit. So it is, if it is detected above the yellow Q, the API manufacturer should develop a strategy to ensure that nitrosamine level remain within the AI limit, remain within the acceptable intake limit. That you have to ensure by making a strategy. Manufacturer should develop an appropriate control strategy, which should include specification limit to ensure that nitrosamine level reliably remain well below within the AI limit in the API. Also, given existing uncertainty regarding nitrosamine impurity and their presence in a drug for APIs, with the impurities detected above yellow Q or at risk APIs, testing of each batch or release should be conducted. So they are suggesting to conduct the testing on each batches if the nitrosamine impurity present above the yellow Q level and below the AI level in your APIs. If it is below yellow Q, you can skip this test for the different batches. But currently, based on a guideline, they are suggesting that every batch where nitrosamine risk are present, that should be checked for the presence of nitrosamine impurity. So alternate approach should be supported by sufficient process understanding and evidence of adequate statistical control, and should be submitted to FDA in a supplement prior to implementation. So if you think that your ROS or your analytical method are sufficiently validated to control this nitrosamine impurity at particular level, you can skip the testing of this nitrosamine in your API. But all this evidence need to submit to FDA first, and then you have to implement by skipping this test. Any API batch found to contain level of nitrosamine impurity above the recommended AI limit should not be released by the API manufacturer for distribution unless with prior FDA agreement. The API is needed to prevent or mitigate the shortage of the drug. So they may allow this nitrosamine impurity drugs which contain nitrosamine impurity to be sold in a certain region where that API is life saving, but still you have to provide the timeline to them, that up to which timeline you will going to change your process and going to control this nitrosamine impurity, and that you have to come into agreement with the FDA based on this timeline. So another guideline has been published, and this has been published by the United States Pharmacopoeia. That is in General Chapter 1469, and it is on nitrosamine impurity. They also suggested some potential source of nitrosamine, which we already discussed. Some solvents may contain the nitrosamine impurities, like solvent may contain presence of acid. It may contain dialkylamine. It may be a recover solvent, or it may be a poor quality of water or solvent. Water is also a source for the either nitrating agent like sodium nitrite, or uh, water itself may contain the nitrosamine solvent. Excipient, the excipient used in pharmaceutical formulation may contain presence of nitrite or other nitrosating agent. Drug substance itself may contain secondary, tertiary, or quaternary amine, and they may give rise to the formation of various nitrosamine. Manufacturing process can also form the nitrosamine by using the acidic condition, using the nitrosating reagent and all, which we already discussed. And even in some cases, like in case of ranitidine, it found that drug itself under stability or under stress condition can form the nitrosamine impurities, which need to be controlled. And container closure, various container closure used for the storing of pharmaceutical formulation can cause or can give rise to the formation of nitrosamine impurity. So all these factors you have to consider while considering the control of nitrosamine impurity, and you have to check for this various factor. You have to have a sufficient analytical method or validated analytical method to check the presence of nitrosamine impurity in all these various sources to control it below the AI limit. So USP published a chapter in USP NF 2021 issue 3 to help industry with analytical procedure. to detect the potential presence of this impurity, and as per this chapter, sources of this. Also, in this chapter, they are given 4 different methods to identify these impurities. The based and these methods are based on 2 methods are based on HPLC with LCMS or LCMS you can say, and 2 methods are based on a GCMS, triple quadruple detector. Next is EMA guideline. EMA is European Medical Agency. You can see here, and EMA also published guideline for the nitrosamine impurity. You have to comply with this guideline to sell your products in uh, EU region, that is European region. So this guideline has been published in 2020, and second revision occur in just recently in 15 January 2024. So highlight of these guidelines are requirement specified with regards to the analytical method. These guidelines are mostly in line with the US FDA guideline, which we have previously seen. Most of the things are common in question answer format. They have given this guideline, and the highlight of this guideline is there is clear mention about the requirement of analytical method. So how sufficient your analytical method should be, that they are given. So guideline propose the testing requirement based on yellow Q, that is limit of quantification. So first is, if your limit of quantification provide the minimum level at which the analyte can be quantified with acceptable accuracy and precision, and should thus be used for impurity testing and decision making. So how this drug can be tested is based on limit of quantification, or periodic testing how it should be done, that is based on yellow Q of your method. That is what guideline has been suggested. So first is, if quantitative testing is performed as a routine control, means if you are adding this analytical method as a routine check to your APIs, that means every batch should be tested, then your yellow Q can be equal to or less than the acceptable limit or AI limit. If you are planning to use this quantitative testing uh, in a skip testing, if you are to justify the skip testing, means you have to not you don't have to analyze each and every batch. Like in first option, you have to analyze each and every batch if your yellow Q is equal to the acceptable limit. In this case, you can skip the testing to 3 batches, 4 batches, or every 10 batch can be analyzed, that you have to suggest to them. But for that, uh, condition is your yellow Q of your analytical procedure should be less than or equal to 30% of the acceptable limit based on the AI. And if you want to completely omit the analytical testing of your API for the nitrosamine impurity, then you have to demonstrate the 3 validation batches, and you have to demonstrate that all of them contain nitrosamine impurities below 10% level, and that can be based on yellow Q of your method. So yellow Q of your method should be less than 10%. You have to completely skip the testing of this nitrosamine impurity. Also, they have given some guideline regarding anti-cancer drug itself, because later on it is found that not only certain other APIs can also contain this various nitrosamine impurities and all. So there are some cancer drug also which reported to contain this nitrosamine impurity. So question arises, as you know, cancer drug is itself can be a mutagenic or carcinogenic. So what should be done in this case? So they have given some guideline that if product intended for advanced cancer only as defined in the scope of ICH S9. S9 is for the anti-cancer drug guideline. N-nitrosamine impurity should be controlled according to ICH Q3A and ICH Q3B. This ICH Q3A guideline is for control of impurities in drug substance, and Q3B is control of impurities in drug products. So if it is an anti-cancer drug, nitrosamine can be limit for nitrosamine AI can be relaxed to the level of ICH Q3A and Q3B based on your dose. In addition, limit according to ICH Q3A, Q3B apply to any impurities considered non-mutagenic based on negative result from a well-conducted in vivo mutagenic study. Generally, if you think that nitrosamine impurities, other than the 6 impurities, there are 140 nitrosamine impurities reported in the market after that. First, it is only limited to 6 nitrosamine impurity, which we have seen, but later on, once the research has started, many companies started working on nitrosamine impurities. They found that there are 140 different type of nitrosamine. But later on, it is also found that all nitrosamine cannot be as toxic as these 6 impurities. So you have to support this hypothesis based on a various tests like Ames test. You have to conduct the toxicity study for this impurity and prove that this impurity is not toxic or not toxic up to the certain level. And if API itself is mutagenic at therapeutic concentration, some APIs can be mutagenic at therapeutic concentrations, mainly anti-cancer drugs. This impurity should be controlled as a non-mutagenic impurity according to ICGM-7. Also, like we are discussing that cancer can cause that AI limit is based on up to 70 years of exposure, but many of the APIs we cannot consume up to 70 years. Like some APIs we use only for 12 months, or some APIs we only use below 12 months, or for 1 month, or for 6 days, 7 days. So for that APIs, FD has published, EM has given some lenience based in a limit. That lenience is, if you are using API only up to 12 months, from 0 to 12 months, then your AI limit is 33 into AI. Like for NDMA, it is an 96 nanogram per day, then you have to multiply 96 nanogram per day to 13.3, and then you have to derive the limit. So that limit which comes is more lenient than what we already seen. And if you are using more than 12 months but less than 10 year, then that limit should be 6.7 into AI. So up to this we have seen US FDA guideline. Also we have seen general chapter of USP published 1469 general chapter for the nitrosamine impurity. After that, Health Canada, Health Canada also published a guideline for the nitrosamine impurities. So highlight of this guideline is they have established the AI limit for the several nitrosamine impurity. So up to this, what we are discussing is there are 6 nitrosamine impurities which should be controlled at their AI. But once the research has been started, I already told you there are 140 nitrosamine reported, various nitrosamine reported by various manufacturer. So Health Canada guideline come up with the limit for various nitrosamine impurity based on their understanding, based on toxicity study, and based on some structural activity relationships. So if you can see the list in their guideline, they have started publishing limit for the various individual API. So it is started from allogliptin, amitriptyline, atenolol, azithromycin. So these are like drug product related nitrosamine impurities. Means this allogliptin can itself going to form a N-nitrosamine impurities, or atenolol itself going to form a N-nitroso atenolol impurity, and it should be controlled at various level. They have started giving the limit based on the toxicity potential or some structural activity relationship. So Health Canada guideline, if you can see, beside these 6 nitrosamine impurities which are discussing, they have published limit for various nitrosamine from A to Z. So I'm just scrolling down to see how much API they are considering are capable of forming the nitrosamine impurities. So you can see from A to, it is started from this up to So this is like 5-6 pages list, and from API A to Z can from the nitrosamine impurity based on Health Canada guideline. So from once this Health Canada guideline has published, it is imperative on all the API manufacturer to check the ROS not only for the certain and for the ranitidine and for other drugs, but you have to check the formation of nitrosamine impurity, control the formation of nitrosamine impurity for all the APIs which you are manufacturing based on these guidelines. So all the ROS undergo scrutiny, all the API undergo scrutiny for the formation of N-nitrosamine impurity. First, you have to do the theoretical assessment if ROS is capable of forming the N-nitrosamine impurity. If possible, then you have to start finding the control strategy for it. You have to develop an analytical method for the control strategy or to demonstrate the control of this nitrosamine impurity. And then you have to decide on testing of this nitrosamine impurity in your release specification. So currently pharmaceutical industry check the nitrosamine impurity for all the APIs which are manufactured. So either they develop a theoretical assessment. By theoretical assessment, they prove that nitrosamine impurity cannot be formed through this ROS, or if formed, then they demonstrate it with the sufficient analytical technique that it is controlled below the AI limit which is published. So now after Canada guideline, there is one new terms has been coined. Up to this, we have seen the nitrosamine impurities, which are like NDMA and which have an alkyl group and N double bond O group. So then there is one new terms come in effect. It is called an NDSRI. So NDSRI is nitrosamine drug substance related impurities. Uh, what does this NDSRI means? These impurities share structural similarity to the API, means it is close to the API structure, and or API fragment or chemical structure, and therefore unique to each API. Means whatever you can see by example propranolol. So this is structure of propranolol, and you can see this is N-nitrosopropanol impurity. Here, this secondary amine is replaced by the N double bond O. So it form a N-nitrosopropanol structure. This kind of structure, now we called it a NDSRI, which is nitrosamine drug substance related impurity. It is unique to each API where amine is present. Then amine get replaced by the nitroso functional group and it forms the N-nitroso impurities. So once a study on nitrosamine has started, many manufacturers found out that even API is capable of forming these nitroso impurities itself. And then this discussion has been started that N-nitrosamine impurity related to API, and this term has been coined called as NDSRI. Previously, there is no guideline on control of NDSRI. There is no AI limit because they all are related to the API, and it is difficult to derive the AI limit for these impurities. So up to before publication of this Canada guideline and this FDA guidance on NDSRI, many of the manufacturers controlling it at the lowest level, like lowest level for any nitrosamine impurity is 18 nanogram per day. But developing analytical method at any 18 nanogram per day is also a challenge. That's why FDA and Canada has published some guidelines based on their study, based on discussion with the industry. They come up with the formula to find out the AI for this impurity based on structural activity relationship of the compound. So this guideline is about how to derive the limit for the impurities, for which there is no sufficient toxicological data is available. So you can see NH replaced by N double bond O. Highlight of this guideline is NDSRI often lack carcinogenicity and mutagenic study data typically from animal studies from which AI limit can be determined. The guidance provide recommended methodology for AI limit determination that use structural feature of NDSRI to generate the predicted carcinogenic potency categorization and corresponding recommended AI limit that manufacturer and applicant can apply in the absence of other FDA recommended AI limit. So FDA has published AI limit for several other nitrosamine also, and if your drug substance contain a nitrosamine impurity or NDSRI for which limit has not been published, so you have to follow this guideline how to derive the limit. This guideline is based on a structural feature of the API itself or structural feature of the nitrosamine impurity itself to find out the carcinogenic potency of this nitrosamine impurities. So here they have introduced predicted means they are predicting, not doing the actual toxicity study. They are predicting this carcinogenic potency of this structure based on a structural activity relationship. In this approach, AI limit to NDSRI assigned based on the NDSRI's activating and deactivating structural feature. So we already seen that nitrosamine are causing alpha hydroxylation, and then it is causing mutagenic effect. So which functional group are facilitating the alpha hydroxylation, which functional group are masking the alpha hydroxylation? Based on that, this AI limit has been derived for this impurity based on this guideline. So structural feature that directly increase or decrease the favorability of the activation mechanism or increase the clearance of the nitrosamine or other biological pathway will have corresponding effect on the carcinogenic potency. This predicted carcinogenic potency approach described in this guideline applies to NDSRI bearing carbon atom on both sides of the nitroso group, where the carbon is not directly double bonded to heteroatom. So N-nitrosamide, urea, and other related structure are excluded from this guideline. Additionally, if this guideline predicted carcinogenic guideline does not apply to the NDSRI when N-nitroso group is within the aromatic ring. So N-nitroso is directly attached to the aromatic ring like indole. This guideline is not applicable to this potency derivation. So FDA has categorized NDSRI into 5 different categories, which we call the potency category 1, 2, 3, 4, and 5, for which they have suggested different limit. Like for potency 1, limit is 26.5. For potency 2, limit is 100 nanogram per day. For potency 3, limit is 400 nanogram per day, and for potency 4 and 5, limit is 1500 nanogram per day. So how to derive this potency category? That is the question now. So this is the flow chart they have suggested on how to derive the potency category. So first is you have to check the structure of NDSRI. Like if you can see, this one is called as alpha carbon atom and this one is called as beta carbon atom for NDSRI calculation. So if you have to check the structure and you have to take the help of this potency chart to find out on which category your API NDSRI is based. So does NDSR have any hydrogen on its alpha carbon atom? If your question, if answer to your question is no, then it is potency category 5 and AI limit is 1500 nanogram per day, which is more relaxed than what we are seeing right now. So if your answer is yes, then second question, does NDSRI have more than one alpha hydrogen on or both side of the N nitroso group? So you have to check if NDSRI are having alpha hydrogen on one side or two side. If answer to your question is no, no alpha hydrogen present, then potency category is 5. If answer to your question is yes, then third question you have to ask, does NDSRI have a tertiary alpha carbon? If answer is yes, then again potency categorization is 5. Then answer is, if it is answer is no, then they have suggested some calculation based on a structural feature for that API, and they have given a potency score 4, 3, 2, and 1. That potency score you have to calculate based on the structural feature of this API, and based on that limit has been defined. So how this potency calculation has been done? So this is formula they are given for potency calculation. It is a potency score which based on alpha hydrogen atom uh, present in your NDSRI. Then deactivating feature score, which is sum of all scores for features present in NDSRI. It is based on a functional group attached adjacent to the alpha carbon atom of your nitrosamine or in nitroso functional group, plus activating feature score. It is sum of all the score for the feature present in NDSRI. So there are three factors you have to consider while calculating this score. First is alpha hydrogen score, second is deactivating feature score, and third one is activating feature score. So what is this that we have to see? So what is the alpha hydrogen score? Or so you have to check the alpha carbon atom near to the nitroso structure. So here you can see these two are alpha hydrogen atom close to this, and at this side there is no hydrogen atom present. So 0 and 2 is the score. So if that type of structure is your NDSRI resembles to this structure, then alpha hydrogen score is 3. So here various examples are given for alpha hydrogen atom 0, 3, 1, 2, 1, 3, 2, 2, 2, 3. It is based on alpha hydrogen present adjacent to the nitrosobridity or nitrogen atom to your compound. So this score you have to find out what on this side and what hydrogen atom present on that side. Based on that your score is derived for alpha hydrogen. So here you find out the alpha hydrogen. This one has been found out from this chart. You have to check the structure of your compound and check the alpha hydrogen score of this compound. Then deactivating feature. So here they have given various deactivating feature for the potency calculation, like carboxylic acid group anywhere on the molecule. If carboxylic acid group is present anywhere in the molecule, your NDSRI potency score is +3. If N nitroso group in pyrrolidine ring is present, then potency score is +3. So this kind of various functional group, based on that various uh, potency score, various individual deactive feature score they have given. So you have to plus this score with the alpha hydrogen. Alpha hydrogen we have already seen how much alpha hydrogen. Based on that score we already checked this score. We have to make addition of the score with the deactivating feature score based on functional group which are present. Here various functional group information is given and some example are given how you can identify this functional group. And second point that you are identifying with this. Then third one is activating feature. So if aryl group bonded to alpha carbon atom, this aryl group bonded to your alpha carbon atom, this you can call alpha carbon atom based on our previous understanding, and aryl group if attached to it, then -1 score has been come in effect. And if your beta carbon atom, like this one is alpha and then this one is beta, then methyl group is attached to the beta carbon atom, then potency score is -1. So all these three can be identified, like alpha hydrogen we already seen, deactivating feature we have seen, and now this one is activating feature. You have to plus all this and derive the potency score of your compound. And where it is fall, that potency score like 1, 2, 3, 4, 5, we already seen that. This potency score has been derived from that. So here is one example. There is one nitroso nebuvolol. Nebuvolol is API and its one NDSRI is having this structure. So it contains alpha hydrogen atom, 2 on this side and 2 on other side of the N nitroso structure. So potency score 2 2. So count of alpha hydrogen is 2 2. Then score is here 2 2. Then score is 1. Then deactivating feature you have to check if hydroxyl group bonded to beta carbon atom. Here hydroxyl group, this is alpha carbon and then this one is beta. So here hydroxy group is present on both side of the n nitroso impurity on beta carbon atom, then your potency score come is 2 plus 2, and 5 consecutive non-hydrogen atom on both side of the acyclic n nitroso group, not more than 4 atom in each chain may be in the same ring. So here they have suggested this structure, and based on that potency score is plus 1. So you have to uh, calculate the potency score from these various factors: count of alpha hydrogen, deactivating feature, and here no deactivating feature present in this structure. There is no methyl group on beta carbon atom. That's why there is no deactivating feature present. So your potency score is 1 plus 2 plus 1, that is come to 4. So potency category is 4. So AI limit is 1500 nanogram per day for this structure. So you have to control this impurity. Instead of at 18 nanogram per day, you have to now control it up to 1500 nanogram per day, which is more lenient and it is easier to develop an analytical method for these impurities. Another example is of propanol. So here n nitroso feature available. So this one we can call the uh, alpha hydrogen or alpha carbon atom on which hydrogens are present. So it is 1 on one side and 2 on other side. So put in alpha hydrogen score is plus 3. Then hydroxy group, one hydroxy group is present on one side, so potency score is 1, and there is no activating feature present, so 0. So potency score is 3 plus 1 4. So AI category is 4 and CPC AI is 1500 nanogram per day for this impurity. So these are more example based on your potency chart, like what we have seen already, that flow chart and decision making chart. So does n nitrosamine have any hydrogen atom to its alpha carbon atom? You have to check this alpha carbon atom. If no hydrogen atom present, then answer is no. Then this structure has to be in category 5. So its AI limit is 1500 nanogram per day. So if yes, does n nitrosamine have more than one alpha hydrogen atom on one or more both side of the n nitroso group? If answer is no, like this structure, you can check n nitroso here and how much alpha hydrogen present on the adjacent structure. If answer is no, then AI category is 1500. Does n nitrosamine have tertiary alpha carbon atom? If answer is yes, like this structure, then AI limit is 1500. For other, we already check how you can calculate the potency score. So based on this potency score, AI limit has been derived, and you have to develop analytical method based on these various AI limits. So currently you have to check all the API for the formation of this n nitrosamine impurity or possible structure. You have to determine based on your ROS that how this n nitrosamine can be formed. You have to give the theoretical assessment to FDA, submit it to them that these are the theoretical assessment. It can be formed, it cannot be formed. If formed, what are the structure? If structure is identified, then what is the potency score that you have to submit? If you have submitted potency score, you have to synthesize this n nitroso NDSR impurity, and you have to submit the control strategy like analytical method, its LOQ, and its strain data in your various batches. You have to submit to get the approval to sell the APIs, sell the API to the manufacturer, drug product manufacturer. So these are various regulatory publications from which I have taken this data. And next question is, as all of you are from the academic background, I believe. So how academic can get involved in this nitrosamine saga? Because from last 5 years, all the pharmaceutical industries are turned upside down because of this nitrosamine impurity, and everyone is talking about nitroso. So first question, while submitting for the approval to FDA, is presence of nitrosamine impurity, absence of nitrosamine impurity, and all that. So academic can also help in this. So first is how you can help. You can help by synthesizing the NDSR. As you can see, all the APIs which having secondary, tertiary amine structure can form the theoretically, at least theoretically, can form the NDSRI. So you can synthesize these various NDSRI, and you can take up the project to synthesize this NDSRI and can submit its standard data to industry. Also, you can develop the analytical method of these individual APIs for the determination of NDSRI at the level which you have calculated. Also, you can study the toxicity of this nitrosamine impurities and NDSRI. As you can see, there is not much data is available on the toxicity study of the various NDSRI. You can take up that study to find out the toxicity study of this compound. Generally, AIMS test is performed to find out the toxicity for this NDSRI and nitrosamine impurity. Also, you can publish the theoretical assessment of possible NDSRI formation. You can check the ROS which are published in various literature. You can check for the theoretical formation of the NDSR and you can publish the document. Uh, it can be a good publication. Also, you can do the potency calculation. You can take up the project, and then uh, you can find out the potency of each NDSRI, which are available in public domain. Also, you can start conducting the AIMS test, and you can derive the toxicological data. So I hope you get to know about what is the nitrosamine impurity and what is currently happening in industry regards to the nitrosamine impurity. This is just a brief introduction, and you can always go through various guidelines which I have suggested to get the in-depth knowledge of the N-nitrosamine impurity and how it is an important aspect currently with the pharmaceutical industry. So thank you very much.
Thank you, sir. Delegates having questions, you can text your questions in chat box. Okay, we are having questions in chat box. Dr. Shahare sir asking, what about nitrosamine impurities in drug products from the sources other than API contamination, sir? Hello. Yes, sir.
So I hope I am able to introduce all of you to this new aspects, importance in a pharmaceutical industry.
Yes, sir. Sir, one question is there. May I read again, sir? Connection.
I think sir is facing network issue.
Hello.
Hello. Sir, one question is there from Dr. Shahari, sir. What about the nitrosamine impurities in drug products from sources other than API contamination?
There is always a possibility that nitrosamine impurity can be from in drug product, that is through interaction with the excipients. Some excipient may contain the nitrite source that can form the nitrosamine impurity, and it need to be checked. That FDA has guideline given that in drug product you have to check for the possible formation of the nitrosamine impurity. You have to do the theoretical assessment, excipient interaction, drug excipient interaction, interaction with the your uh, packaging materials and all, and you have to control it accordingly.
Okay, sir. One more question is there, sir. How to avoid nitrosamine impurities, if any, in potable water? Are are there any preventive measures on daily basis?
There are two things you can do. Uh, from potable water, there are two sources. Like potable water can contain sodium nitrite, which may further react with uh, your APIs, or can react in your ROS, various solvents you are using. Like it is like a nitrosating source for the formation of nitrosamine, that sodium nitrite present in water. So currently, what industry is doing, they have developed a method on ion chromatography to find out the nitrite ion present in a potable water. They are defined certain limit, and they control it in potable water, whatever they are going to use in manufacturing of API. That below that limit, that potable water should contain the nitrite. And if nitrosamine are present itself in a potable water, there are various analytical methods available on various forum, that you have to check for the potential source of nitrosamine.
Okay,
So
sir.
two things you
Thank you
can
very
do:
much.
first check the nitrite ion, and then check the nitrosamine through various analytical methods. There are generally
Okay.
GCGCMS or LCMS method easily available.
Okay, okay, sir.
So industry
Okay, sir.
currently doing by identifying the nitrite present in water to minimize the formation of nitrosamine through reaction.
Okay, sir. Sir, as there are no more questions, I think that was all about it. I would like to thank you
On
on
lighter
behalf
note,
of Sir MSG Copper. Yes, sir.
on lighter
Uh,
note, I can say that in even wine, uh, nitrosamine can be present. So you can be careful while drinking the even wine also.
okay,
Right?
sir.
You are in Nashik,
Okay.
so I must say this. Okay.
Thank you, sir.
So on lighter
Thank
note,
you.
don't take it personally. Just I'm saying.
Yes, sir.
But
Yes, sir.
fermented product can form the nitrosamine, so it can be possible. Yeah.
Okay, sir.
Okay, ma'am.
Okay. Thank you, sir, for giving insights on nitrosamine impurities. Thank you so much. Now I request to you, please accept the certificate as a token of love on behalf of our college.
Thank you, ma'am. Thank you for invitation, and I hope I can ignite some curiosity about nitrosamine impurities in all of you, and you people
Yes, sir.
can
Yes,
start
sir.
working on it. Yeah. Thank you.
Yes, definitely, sir. Thank you once again. Dear delegates, we are going to start the next session sharp at 2 p.m. The link has been already shared on the group. So, thank you very much.
Can I leave?
Thank you, sir. Yes, sir. Yes, sir.